pour savoir jusqu'à quel point aujourd'hui c'est utilisé dans la sélection variétale actuelle.
Alors, nous avons ici le spécialiste, n'est-ce pas, qui travaille sur le pommier, mais je vais répondre et puis il complétera si nécessaire. The marker-assisted selection is a very new promising tool already applied in the different species I talk about. It's already applied for apple to follow the different genes of resistance, especially for scab resistance. So we are doing this type of work here in Angers. It is also used for the resistance to Melodogine in Prunus rootstock by incorporating some quite interesting genes. And it is also in progress in apricot for the Charca resistance. So again, you know, this is new ways, very promising, already tested, not only tested but used. Now we have to you know, we did the first step, the second step. Remember what I explained. We selected in the greenhouse, and we have to validate in the near future in the orchard.
Could you say something about the generation time? Then you can very much shorten down the cycling of new varieties.
Yeah. Yes, this is first of one, to be more efficient. You know, to be more efficient. For example, to incorporate several constructs to get what we call the durable resistance. So that's the first interest, because by standard crossing, by not using the markers, these flags along the chromosomes, we have to do another generation to check if we have the right genes. Thanks to this technology, we know readily at the seeding stage that we have certain genes, and that we maximize or we minimize the risk of apparition of a new race. So this is the main interest. We will be more efficient, but using just this technology, we have to wait the juvenile period, because a seeding is juvenile, and we have to check at the orchard the incorporation of the right genes, and to wait to get the tree, to get the adult phase of the tree.
Oui, François Colson, Agrocampus Ouest. Deux questions. La première, c'est dans le cadre d'ISAFRUT, cette coordination européenne permet-elle d'avoir, je dirais, des progrès significatifs par rapport à d'autres recherches dans d'autres parties du monde? Est-ce qu'on avance plus vite en Europe sur ces questions-là qu'aux États-Unis, par exemple? Et puis la deuxième question, c'est en matière de qualité des fruits gustative et résistance aux maladies. Est-ce que les deux facteurs peuvent aller dans le même sens, ou est-ce que parfois on peut avoir contradiction entre une meilleure qualité des fruits et une plus faible résistance aux maladies du fait de la protection de la cellule?
So, to the first question, you know, of course, European projects are a fantastic tool to improve our European efficiency. You know, this is a very important concern, and we are, especially, well, in the different species, but especially on apple, we are working now for a long time on cooperation for non-competitive research, of course, for example, for locating the gene of interest. So we are doing this work now for a long time, and Isafut is a very good example of how the work we did before is now put at the same level in different teams in Europe. Yeah, this is very important, and our next project, I hope, will be on this topic, especially on this topic, to improve our breeding efficiency in Europe by sharing the pre-competitive work and by selecting new material thanks to the markers-assisted selection to breed with a more efficient way, in a more efficient way, new variety, not only for the public institutes, but also for the private companies, which are more and more interested in breeding new varieties. Now, about the second question, there is no evidence of links between quality and resistance. Of course, if you find a gene in a wild species, of course, in a gene of resistance in a wild species, the quality of the wild species will be poor. But now, with the material we selected in the previous years, we have at a very good level of quality resistance genes which are in our new varieties. We have an example. At lunchtime, we see a new generation of apples which are less susceptible to diseases, and there is at this stage you have for most of our genes independency between quality and resistance, and we can progress both for quality and resistance.
One last question.
It's not a question, just a comment to okay. Add something to what Yves told about François' question about the competition between Europe and the other countries. Actually, for me, there are two different kind of collaboration, or there are two different kind of situation. There are the research part, where actually we're working together with New Zealand, the US, and other part of the world, because the science is very expensive, so it's better to share the tools and the advance. And there's the breeding part, the competitive part, where we are still competitors. So I would say that for the next proposal we are working with the other countries outside of Europe for science, but we will be still competitors for the breeding.
Thank you, and thank you, Yves, for your answers as well.